with that, we're going to move on to the last speaker of our session, which is Professor Sandrine Betuing, who's a full professor at Sorbonne University in Paris. Sandrine did her initial training in Toulouse, had a postdoc at Harvard Medical School, and came then back to France. And she's now a full professor at Sorbonne Université since 2018. So Sandrine, please.
Okay, can you hear me? Can you see my screen? Okay, so thank you very much for the invitations. I'm very glad to be here today. And I'm going to focus my talk on cholesterol metabolism as a therapy for Huntington's disease. So as you may know, a mutated Huntingtin can act through several cellular dysfunction, and especially transcriptional dysregulation, endoplasmic reticulum stress, mitochondrial dysfunction, as well as mutated huntingtin is able to impair trafficking of mitochondria and trophic factors. And we can see also altered neurotransmission, and especially glutamate glutamate release is altered from cortical synapses. And we can see also aberrant synaptic signaling, and especially calcium dysregulation. For the all mechanisms are involved in neurodegeneration. But we know also there is so many papers related to glial cells, and we know that neuroinflammation from microglia, but also from astrocytes, is involved also in HD pathogenesis, as well as a deficiency in myelinating. So this is the whole picture that we know about the cellular consequences of expression of mutated Huntingtin. But from the last decades, many papers have been also published on cholesterol metabolism. And so alterations of cholesterol metabolism starts with Elena Cattaneo's team in Italy from Milan University. And we also focused on this cholesterol metabolism in the team. So why cholesterol metabolism is interesting to study? First, because the brain, in the brain, we have 23% of total cholesterol is from the brain. And cholesterol is very important as a structural component in plasma membrane, and especially in lipid raft domain, because it regulates intracellular signaling. And most of the cholesterol, 70% of the cholesterol you will find in the brain is involved in myelin. It is the main component of myelin. But cholesterol is very important also for vesicle biogenesis and for synaptic transduction. Okay. So this means that if cholesterol metabolism is altered, so it could influence on cellular functions. And especially we know in the literature that when cholesterol metabolism is altered, it impairs generation of synaptic vesicles. The distribution of receptors within the plasma membrane, and especially in lipid raft, is also altered. And basically it reduced neuronal survival. So what about cholesterol homeostasis in the central nervous system? So cholesterol cannot cross the blood-brain barrier. So it means that cholesterol synthesis is synthesized directly within the brain. And in adult brain, glial cells, and especially astrocytes and oligodendrocytes, are able to synthesize cholesterol. So cholesterol, when it is synthesized, it can move to neurons thanks to lipoprotein, and especially ApoE bind lipoprotein, and then the cholesterol can then enter in the neurons. And the turnover of cholesterol is due to one enzyme that I'm studying, the CYP46A1 enzyme, which is a neuronal enzyme. And this enzyme is able to hydroxylate cholesterol in 24-hydroxycholesterol. And this metabolite can cross the blood-brain barrier to be excreted in the periphery. So what about cholesterol homeostasis in HD? So it has been shown for the last decade that this cholesterol homeostasis is dysregulated. And this is a very summarized picture of the synthesis of the different pathways to synthesize cholesterol. And it has been shown that decreased cholesterol precursors and synthesis enzyme in HD, so there is a downregulation of both the enzyme, several enzymes, and the precursor, especially lanosterol and latosterol, are downregulated. And also the product of the enzyme I was talking about, the CYP46A1. So the product, the 24S hydroxycholesterol, is decreased in patients' plasma. And this decrease is correlated with the caudate atrophy in patients. We are focused on this enzyme, the CYP46A1, and we show that the expression in patients' putamen is decreased. And we also confirm that in model mice, and especially in the R62 mice at 12 weeks old, we can see a decrease of expression of CYP46A1, either in cortex and in striatum. So we made the hypothesis that maybe a restoration of this enzyme could restore the old cholesterol metabolism and maybe may have an impact in HD mice. So to study the role of CYP46A1 in HD, we first restore its expression in a cellular model of HD, so in primary striatal neurons, where testing a mutated huntingtin, so you have a decrease of surviving of neuron expressing the mutated huntingtin. And when we transfect the cells with CYP46A1, we are able to alleviate the death of neurons. So we have an increase of surviving neurons. As well, we have also a decrease of aggregates formation. So mutated huntingtin, as you all know, is able to aggregate, and CYP46A1 can decrease this process. So then we did some in vivo study on R62 mice, and we, with a gene therapy approach, we restore expression of CYP46A1 directly within the striatum of the mice. So using an adenovirus carrying the coding sequence of CYP46A1. And by doing that, we were able to improve motor and neuropathological phenotype. And as illustrated here, rotarod performance, you can see here on different groups of wild-type mice, and you have here the R62 mice. We were able to alleviate motor dysfunction on rotarod when mice are infected with an AAV with the CYP46A1 sequence, as you may see here in blue. So we also look at aggregation within the stratum. And as you see here on R62 mice, you can see aggregates, mutated huntingtin aggregation. And there is a decrease of aggregation with CYP46A1 restoration. So we wanted to go further on the mechanisms that were able, who could explain how CYP46A1 is neuroprotective in HD mice. So for that, we use the ZQ175 mice. We show many dysregulations related to transcription, to motor impairment, excitotoxicity. These mice show also impaired energy metabolism. So we use these mice to go further and to explain why CYP46A1 could be a neuroprotective in HD. So we first show that the CYP46A1, the messenger, as well as the protein, is downregulated in the striatum at 7 months old, but also you can see that this decrease is more severe with older mice. And we use exactly the same strategy as we did before with the R62 mice. So we restore CYP46A1 expression directly within the striatum using AAV. And we did that at 4 months of age of the mice. And then we evaluated behavior, motor behavior, during several months. And then we sacrificed the mice to study cholesterol metabolism, gene transcription, as well as neuropathology. So first, I will show you results from motor dysfunction. So we evaluated motor dysfunction using cyclotron to have access to a locomotor activity. And we also studied rearing frequency and the rotarod. And basically what you can see here, so we have heterozygous mice result on the upper panel, and just below you have homozygous results. So what we can see in the blue line, this is heterozygous with CYP46A1 restoration. As compared to wild type, we can see that we are able to restore locomotor activity as compared with the heterozygous mice, the control heterozygous mice. And we had the same conclusion with the rearing frequency and as well with the rotarod performance, especially for homozygous mice, all along between 7 months to 11 months of age. Then we looked at neuropathology, and first we looked at soma atrophy within the neurons of the striatum. And basically what we see is that in the heterozygous mice, there is a decrease of striatal neurons area. And we were able to alleviate this atrophy with CYP46A1 expression, either in heterozygous mice and in homozygous mice. What about aggregation? So for aggregation, we saw also a decrease of aggregation number as well as aggregate area when CYP46A1 is restored both in heterozygous and homozygous mice. So, So basically for neuropathology, CYP46A1 is able to improve motor dysfunction as well as neuropathology.
Sandrine, 1 minute left, please.
Yeah. So then we looked at cholesterol metabolism and especially the precursor of metabolism, and we were able to see that CYP46A1 is able to increase the synthesis of cholesterol precursor. And then we look at transcription, and to make a long story short, we are able to see that CYP46A1 is able to restore a numbers of genes that are downregulated in these mice, and especially so we were able to see major pathways regulated by CYP46A1, and especially neurotransmission, synaptic activity, cytoskeletal dynamics, autophagy and proteasome, as well as regulation of innate immune response. And I just want to illustrate the neurotransmission and synaptic activity we were able to see that CYP46A1 is able to increase spine density and also to increase synapse density using two markers of the pre and the post synapses. And also electrophysiological studies that CYP46A1 is able to improve the frequency of excitatory postsynaptic neurons. So basically, to sum up the whole picture, what we think is that by restoring CYP46A1, especially in neurons, we are able to reactivate cholesterol synthesis in glial cells and especially in astrocytes. And this is associated with a global transcriptomic signature associated with a better synaptic transmission. And I didn't have the time, but we also restore autophagy. So to explain that, so now we need to go further to explain how CYP46A1 can modulate transcription. And we have an hypothesis through the liver X receptor, which are nuclear receptor, and some precursor of cholesterol like desmosterol is a ligand for this receptor, as well as the 24S hydroxycholesterol is a ligand for it. So I want to thank all the people involved in this result, especially the PhD student Rabia Kasher and Colleen Mounier. Maybe you have seen her poster in a EHDN meeting. And I want also to thank all the fundings and especially EHDN funds. Thank you.
Thank you very much, Sandrine, for this really interesting talk.